feed the world, the same staple crops are now grown in every continent: rice, wheat, potatoes and maize. If these crops failed, we would all be in serious trouble. We now know that each crop actually originated in specific areas, centers of diversity where the genes we may need for future crop breeding may be found. This concept of centers of origin was first proposed by the great Russian scientist Nikolai Vavilov. Today, more than 50 years after his death, the significance of his pioneering work is undiminished, for us and for our children.
Nikolai Vavilov Institute of Plant Industry in St. Petersburg, named after its most respected director, one of Russia's greatest scientists. Nikolai Vavilov made the first comprehensive collection of crop varieties from all over the world for study here at the Institute. He dedicated his life to the development of science for the benefit of society, to improve the food supply for the people of Russia. He travelled tirelessly to many parts of the globe, searching for samples of plants and seeds for use in plant breeding. He has inspired generations of crop breeders.
Nikolai Ivanovich Vavilov, senior scientific worker, former member of the Academy of Science of the USSR, arrested 6th August 1940 and charged with betrayal of the motherland and spying. Interrogated by Comrade Khvat during 1,700 hours up to July 1941, found guilty by the War College of the Supreme Court of the USSR and sentenced to death by order of the great leader Joseph Stalin, transferred to Saratov jail in disgrace, after 2 years moved to the prison hospital, died June 1943.
Vavilov's name has now been restored, and today his portrait hangs in many scientific institutes all around the world. Some crop scientists still remember him as a colleague and friend.
Yes, well, I met him in 1938. Vavilov came out from his office, greeted me with great enthusiasm and friendship, and I was overwhelmed. He treated me like a learned colleague all the time. Very, very outward going, extremely friendly, talked very good English, and had a really worldwide knowledge of plant genetic resources, or plant diversity as it then was. He was a very bright young man, as you can imagine. After doing his ordinary degree, he registered for what we'd call a PhD, and was during that time given some money to go and visit other countries. So he came to England and he went to the John Innes Institute and met Sir Daniel Hall, the director, so that he began to understand what the geneticists and plant breeders were thinking at that time. He was a man in a million, I should say. I've never met anyone like him before, and I was extremely impressed by him, and still am to this day.
It was just these personal qualities and love of life that endeared him to scientific colleagues and ordinary people alike.
He made his first collecting expedition to the Caucasus, and he was absolutely amazed by the amount of diversity within the crops they were growing. Crops that they knew in Russia and in the rest of Europe to be rather non-diverse, very simple, just a few varieties. Out there in the Caucasus, he found an enormous amount of variation within the crops themselves, far more than anyone had realized existed before. And he also found a lot of wild relatives of the crops. So this started him off in his quest for plant diversity, genetic diversity as we now call it.
From the First World War to the late 1930s, Vavilov's travels took him to many parts of the world and allowed him to build up a unique knowledge of the local varieties or landraces of many important crops. He came to realize that certain areas had far more diversity than others. He called these the primary areas of origin. They were mostly mountain regions in the tropics or subtropics.
So this was quite new. No one had, first of all, realized that there was such a lot of diversity amongst the crops in their centers of diversity, which very often were the centers of origin, and no one had realized that to look for resistance to plants and diseases of these crops, you had to go to where these crops had evolved over the millennia with the plant diseases or the insect diseases or whatever it was, and had evolved some kinds of resistance to them. So that was the genius of Vavilov. He was enthusiastic about that. He passed his enthusiasm to many other colleagues and to youngsters like myself.
The seed samples that Vavilov and his team had collected around the world were stored in metal boxes as the first major international gene bank. On the Institute's experimental farms, the seeds were used in plant breeding to improve Russian crops. After all, this was Vavilov's fundamental aim, to use science to alleviate hunger and to improve the lot of the Russian people. Specialist teams began long-term programs with a number of crops such as cabbages, onions and cucumbers. This was an ambitious program. Crop improvement takes years to succeed. Although they had already started producing improved seed for use in Russia, their work was increasingly hampered by political attacks on the Institute and its director.
The main protagonist of the camp which criticized Vavilov's work very much was a man called Trofim Lysenko. I met Lysenko the first day I was there, and although I was a young man, I could see that that man was a troublemaker. I said to Vavilov, you know, that man is dangerous, you should watch him. And Vavilov said, because he was such a nice person, he enjoyed everybody, he said, well, he's a young man with ideas, he ought to be allowed to develop them. Well, that man did develop his ideas, which were completely unscientific. They were very much along the line of the communist government, because this man said that you could change anything by changing the environment, including the genetics of plants and the genetics of man and all the rest. He was very critical of formal genetics, as he called it, Mendel and all the other geneticists. He felt that this was a pseudoscience and had nothing to do with reality. Of course, he was absolutely wrong. But he got the ear of the communist bosses, Stalin particularly. So more and more funding, more and more facilities were made available to Lysenko and fewer to Vavilov. Lysenko criticized Vavilov to such an extent that he was then considered to be by the communist government a spy, a capitalist spy, and all the other terms that they used for people they didn't like.
With Nazi Germany now at war with Russia on the Eastern Front, Vavilov set off on an urgent field expedition to the Ukraine. He wanted to collect the local land races of wheat and vegetables before they were destroyed by Hitler's advancing troops. But the political authorities had already decided to stop Vavilov's work. Without waiting for him to return, the secret police were sent out to find him. He was arrested, brought back to Moscow and thrown in jail. No one at the Institute in St. Petersburg was told a word. Vavilov's desk remained empty. Within days, other senior staff were taken away. Those remaining feared the worst.
очень дорожили этим именем и их идеалом и в науке, и во всех.
Of course, there was a lot of talk about what had happened to Nikolai Ivanovich, what his fate was, and it was still unclear where he died, how, and in what circumstances, so people were very concerned. But there was only outside or at home. It couldn't be mentioned within the walls of the institute.
Переживали люди в этом плане. Ну, это вот в пределах домашних, но не в стенах института.
The problems at the institute soon paled into insignificance as the German advance reached St. Petersburg, or Leningrad as it was then known. The city was under siege, and the whole population were in fear of their lives. In the first winter of the siege, food supplies dwindled to nothing. Many who had survived the bombing now died of starvation. The institute was officially closed down by the government and many staff evacuated. Those that were left struggled on as best they could. They knew that the seeds in Vavilov's gene bank could have been used as food. But Vavilov had instilled an extraordinary commitment in his staff. Not a single grain from the collection was touched.
Я еще раз хочу сказать о том, что люди были настолько, я думаю, что
I think that it was the spirit of Nikolai Ivanovich. Everything was founded by him. He selected the people for himself, who were really dedicated to the work. They lived by the spirit of Nikolai Ivanovich, so that the collection was saved for science.
But at a cost. Several senior scientists collapsed and died at their desks. The germplasm they saved is still used in crop breeding today. Meanwhile, Vavilov suffered in prison, vilified for his independent ideas, his open mind, the inconvenience of his scientific convictions. Starving in his cell, Vavilov's whereabouts were unknown to his colleagues and even to his wife and children. No one knows the exact details of his last days.
В январе 1943 года мы об этом узнали только.
We knew nothing. My mother only found out he was there much later, in 1943, and she didn't tell me about it, so as not to upset me too much. I didn't actually find out that my father had died until a year later.
Even the whereabouts of his grave are unknown. His death was kept secret for years, but his ideas and ideals live on. With Vavilov in prison, it was Lyshenko, Stalin's man, who took his place and soon dominated the Academy of Sciences. All studies in genetics were halted. Russian agriculture and crop research declined under bogus direction and conflicting political dogmas. When Lyshenko was finally dismissed with the fall of Khrushchev, the country was starving. Food production was less than it had been in the 30s. The true value of Vavilov's pioneering work has become increasingly apparent. Without his foresight, thousands of unique landraces would have disappeared completely.
Landraces, it is native aboriginal varieties, very ancient. And Vavilov and his co-worker very carefully collected these varieties. And now, when we have new methods of breeding, like gene engineering, the genes of landraces begin to be very important for increasing of adaptivity, for increasing of tolerance, for increasing of yield, for increasing of quality of the yields. Because in all world now, all people who work in the field of plant genetic resources, they begin to understand that genes of land grasses, it's most important genes for the future breeding process.
Vavilov was the first to demonstrate the true extent of our global interdependence, but its significance was largely neglected at the time. Then, in the 1950s, fears were raised internationally about prospects for the global harvest. Politicians became concerned that a hungry world would soon be a more dangerous one. One of the first research efforts to make use of Vavilov's approach to plant genetic resources was sponsored by the Rockefeller Foundation in Mexico. In the early 50s, Dr. Norman Borlaug led an international team of scientists charged with finding ways for farmers in the developing countries to produce more food. With so much of the world's grain being grown on poor soils, their idea was to increase yield by using artificial fertilizer. The problem was that this didn't work with existing seed. The plants grew too tall and collapsed under their own weight. Like Vavilov, they started collecting land races. For maize, they concentrated on Mexico, which they knew was its area of origin. But for wheat, whose origin is in the Middle East, samples were obtained from collections made in Syria and Turkey. Seeds from the Caucasus and Central Asia were obtained from the Vavilov Institute. The real breakthrough actually came from elsewhere. Japanese scientists sent some short-stature landraces called Norin. Adding Norin genes to the crossbreeds they'd already produced, Borlaug's team bred new dwarf varieties which showed remarkable genetic potential.
It wasn't just the new high-yielding varieties in India or Pakistan in the original Green Revolution. Those varieties had to be cultivated in the right way. They would only be marginally higher yielding under worn out soils. They had to have food, which we call fertilizer, to restore worn out soils in order to express their high genetic yield potential. And there were a lot of other things. You had to plant them at the right time, the right seeding rate. You had to get the weeds out at the right time before they sucked up all the fertilizer. And when all of those pieces went together, and these young people who believed in it demonstrated this out on thousands of farms. It caught on, and it surprised everyone. We call it a Green Revolution. We didn't put that label on it. That was done by, I think, Dr. Dowd of the USAID, when he said, look, something is happening in India and Pakistan. A few years ago, there was no hope, and now it looks like a green revolution.
Under intensive irrigation and with subsidized fertilizer, yields in the Punjab doubled and trebled. Food production rocketed. For the first time, India faced not famine, but surplus. The effect was immense. The so-called miracle crops were front-page news. Politically, they were seen as contributing to world peace. As team leader, Norman Borlaug was awarded the Nobel Prize for Peace in 1970. The science of plant genetics had emerged from scientific obscurity to the global spotlight. Food production was booming. Plant breeders enjoyed their finest hour at home and abroad. Farming had become big business, with seeds and agricultural chemicals supplied to the farmer in a single package. Both production and profits soared. Throughout the 1960s, just a few high-yielding hybrids blanketed the Corn Belt. In fact, they were very closely related to one another, and all shared particular inherited factors. The industry had forgotten that constant struggle between plants and pathogens that genetic diversity provides for. Production was high, but the genetic diversity of corn had become dangerously low. A new disease appeared in the damp summer of 1970. Southern leaf blight. Farmers found that fields of corn could die overnight. The whole corn belt was threatened with catastrophe. But the weather gradually improved. The problem was contained. Estimated cost, $150 million, and it could have been much worse. Corn breeders started a frantic search for resistance to the new disease. They soon discovered that the weakness was due to a single genetic factor. So seeds of varieties without this factor were urgently multiplied in other parts of the world to be rushed out to corn belt farmers in time for the next season. A total disaster had been averted, but a new term had been coined: genetic vulnerability.
Southern corn leaf blight that devastated the U.S. corn crop in 1970 became the subject of a significant study by the National Academy of Sciences that was published in 1972. And what the Academy discovered is that the narrowness of the genetic base that appeared to be responsible for the southern leaf blight in corn turned out to be a problem that wasn't unique to corn, and they found that it was common in many other crops in the U.S. Their basic conclusion was that vulnerability had moved from being a strictly U.S. problem to being a global issue and a global problem, the potential severity of which they really couldn't estimate.
The American government report came just in time for the representatives of the 120 governments who met in Stockholm for the world's first Earth Summit.
At that time, agreement was reached on these points: that the sources of genes were very important for humanity, and that to preserve them forever was a mission for the human race. So, subjects which previously had been discussed only by specialists reached the level of the United Nations. And because of this, there was a consensus formed, not only among scientists, but even among politicians, about what had to be done.
Some countries already had their own collections, and it was decided to set up a network of publicly funded international gene banks through the Consultative Group on International Agricultural Research, known by its acronym, the CGIAR. Wheat and maize were already being conserved in the gene bank at Borlaug's Research Institute, the International Maize and Wheat Center near Mexico City. Now, all the world's major food crops would be provided for. In Vavilov's time, the samples were stored at room temperature in varying humidity. As a result, the seeds often deteriorated after a few years. Seed storage methods have improved since then. Today, samples are dried to a standard 8% moisture level and then vacuum packed before transfer to a cold store. This is the gene bank at the International Center for Research in the Dry Areas, or ICADA, in Syria. It houses thousands of wheat and barley samples and their wild relatives, together with a variety of other crops like lentil, faba bean and grass pea, and some fodder species like vetches. Gene banks such as this one set the standards for seed storage today and set guidelines on all aspects of gene bank maintenance. Today, CGIAR centers hold over 500,000 samples of crop plants on behalf of the world community under the auspices of the Food and Agriculture Organization of the UN. The collections are available to users everywhere without restriction. They represent global insurance of the world's crop diversity that has developed over the millennia and is part of the common inheritance of humanity. Vavilov would certainly approve. But seeds are living germplasm. Some of them die, even in cold storage, so there always has to be an ongoing cycle of testing. Just a few seeds are taken out of each gene bank sample and sent to the germination room. After a few days, in warm and humid conditions, the germinated seedlings are counted. If any sample shows more than 15% failure, then it has to be regenerated by growing the plants. So every gene bank needs substantial backup resources. A large farm where samples can be grown out and the seeds collected. Better still, a network of farms in different climatic zones. This requires a lot of money every year. The Kada Gene Bank holds over 8,000 samples of common lentil. Here some are being harvested. Not only the suspect samples, sooner or later every sample has to be regenerated before it deteriorates. Just part of the necessary cost of maintaining the world's food security. The genetic resources held in international gene banks are in constant use. Plant breeders search for genetic characteristics like disease resistance and drought tolerance. Bred into new or existing lines, these characters are then made freely available to plant breeders everywhere, so they can be used to improve crop varieties. Since the beginning of agriculture 12,000 years ago, farmers have saved their seed for next year's planting. Now, more and more farming communities are setting up their own gene banks, and links are being developed between farmers' groups and both national and international gene banks.
What's happening in the communities when farmers maintain their varieties? They maintain not only the germplasm, but also a lot of knowledge. Collections of germplasm by gene bank people separates this germplasm from the associated knowledge, and little, if any, of this knowledge is recorded and entered into the passports. And when you don't have this information, it's very difficult to know what to use and what not to use. We need farmers who are willing to still maintain diversity in the traditional way and maintain them in an evolutionary way and maintain the knowledge and continue studying, observing what they have, so that knowledge about these can exist, continue to exist and continue to be available in farm communities, because this is a major repository not only of germplasm but also about vital knowledge.
This one really is good. Has quite some seed. There's a lot of clover here.
Here, a small team from the International Livestock Institute and the Ethiopian Biodiversity Institute are visiting farmers to examine the wild legumes growing in the pasture land and to collect samples.
Inside, in the hay, this trifolium presence is essential for livestock feed.
Yeah.
That's what he commented.
So they care for it.
Yeah, they care for it. Yeah. They know the value of the trifolium.
Very good.
Yeah.
Clovers and vetches are important in many pastures as they fix nitrogen and boost the soil's fertility. Today, documentation includes a precise location pinpointed by a satellite positioning device. In Vavilov's day, things were much less precise. He was dependent on guides who sometimes didn't know the local area at all.
Just tries and calculates. Yeah, because I think we share. He shares with us his information, we share with him our information. Okay, then let's distribute some bags and start collecting. Will you help us? We need to collect a real wide range of diversity So we should go and spread all over this place and collect as many different samples as possible to have a full sample of the diversity.
Some species are quite difficult to separate after flowering, but the team will collect 3 different species in separate bags. This is now the recommended technique for modern collecting of germplasm, a real collaborative venture between scientists and farmers, combining high technology with local knowledge. In the past 30 years, many countries have set up their own national gene banks, though few are as advanced as this one at Tsukuba in Japan. Here, over 100,000 varieties of rice and other crops are stored. The Japanese government sets a high priority on the conservation of genetic resources, and Japan is one of the world's largest supporters of international efforts in this field. Here, computers keep track of the different samples, and robots collect them, identifying them by barcode. Samples in use are kept at -1 degree centigrade. Long-term storage is at -10. Under these conditions, rice seeds can be stored for 30 years or more without deteriorating. The few human workers are found in the anterooms, preparing samples for sending out to breeders or other gene banks and extracting seed for germination testing. Today, many gene banks also ensure that samples are kept in duplicate elsewhere, a safety precaution in case material is damaged or lost due to unforeseen circumstances. And around the world, collections are damaged all too often, especially in times of war or other social upheaval. Rwanda's civil war left a countryside that was deserted. Abandoned fields and houses all across what had been one of Africa's most densely populated countries. In these hills, every valley has its own peculiarities. The farmers of these steep slopes were masters of eking out the best from tiny plots. Many villages grew their own unique varieties of all local crops. When the refugees started to return to Rwanda, international aid provided families with small quantities of food grains, mainly sorghum and beans. In the villages, they found some root crops still in the ground. Sweet potatoes and cassava could be harvested to tide them over the first season. The problem was many families had lost their seeds for other crops. Apart from sorghum, the main staple crop in Rwanda is beans. Every family has a plot, and for many people, this is their major source of protein. Different bean varieties are grown together. Every village has its own unique mixture, specially adapted to the local conditions. Before the war, scientists had collected over 650 types of Rwandan beans. These were carefully documented and samples were stored in the National Gene Bank. In the war, the gene bank was destroyed along with the records. The retreating Hutu army gutted the place before they left. Even before hostilities ceased, an international crop recovery project called Seeds of Hope for Rwanda was started. International gene banks all over the world swung into action to restore the local agriculture by providing seed from Rwandan samples held as duplicates in gene banks abroad.
Yes, that's what we're trying to plan now, as to how to make better use of regional resources.
Rwanda is a particular case. You cannot just take any seed from anywhere and bring it to Rwanda to produce it. We went in the road from Kigali to Karama. You see a huge variabilities in the altitude. You go from 1700 to 1400 and then up back to 1600, which means these micro-ecological places will need a specific type of a seed or germplasm.
A number of aid agencies brought seed from other parts of Africa and distributed it at random. Some farmers planted it to feed their families, but they soon had a nasty surprise. When Jean Vianney's sorghum came up, the plants grew all right, but they did not set any seed. It was the wrong variety for his area, and his crop was a complete failure.
A sorghum suited for highland when it is planted in the lowland. Some of them have something like 5 to 10% of seed setting. Looking at it, we can feel like this is an attack by a bird, but it is not that. It is the poor seed setting which will induce a great loss on some of these farms.
Sorghum samples were taken from a CGIAR gene bank in India and grown out in Nairobi, Kenya, where the climate and altitude were similar to parts of central Rwanda.
What this project does is give the opportunity for gene banks to return some of that material to benefit the farmers that donated the material in the first place. And in the long run, this means that farmers in Rwanda can reestablish an agricultural system which is most appropriate to Rwanda, rather than having to rely on imported seeds, which for the most part have been bred for other conditions faced by other people in perhaps faraway lands.
As a result, farmers like Elsie Tsuvanu Kabandi were supplied with the same varieties which she'd been growing here before the war. Scientists and farmers in Rwanda used to lead the world in developing collaborative bean breeding programs. This advanced research was completely disrupted by the war, although the Highland Research Station, where some of the unique germplasm had been collected, survived intact, saved from destruction by the dedication and bravery of some of its staff.
We made up a committee consisting of 6 people, and I was the president of this group. We tried to put men on guard to protect the seeds in the store until World Vision came here afterwards, and we could start our work again. Then the head of the station and other technical people came back and found all the seeds we had been able to protect in our store.
With the help of gene banks elsewhere, especially the International Center for Tropical Agriculture in Colombia, a CGIAR center, the full range of bean varieties could be restored, and even the bean improvement program was soon re-established too. International gene banks have helped prevent famine in Rwanda. But more than that, subsistence agriculture has been restored in just a few growing seasons. The international system of gene banks is a major enterprise, but it's not easy to maintain.
Just the sheer amount of management and record keeping and so on, it can be very prohibitive, particularly in countries with lack resources and human resources, training and infrastructure. For many species, they're very difficult to keep in gene banks, particularly for crops that don't produce regular seeds. Then the options are to keep them in field gene banks or in vitro collections, which means in test tubes in laboratories, which severely limits the amount of material that can be kept.
Bananas and plantains don't usually produce seeds, so conserving varieties is quite a problem. One method is to keep them like this, in a field gene bank. Space is conserved by keeping just 4 plants of each variety planted in a regular pattern. It's essential to keep good records. Some varieties may not be easily recognizable when grown under unfamiliar conditions. Field gene banks have the advantage of maintaining both crops and their pathogens under natural conditions. But there can be major drawbacks. Pests or diseases can get out of hand, and the weather too can give cause for concern, especially here in the Philippines. A single tropical storm could wipe out the entire collection, so they are now developing a backup facility in their laboratory.
How are the cultures today? We're trying to maintain a duplicate of all the accessions being maintained in the field gene bank. We're trying to maintain it in vitro. It's a backup, actually, backup gene bank, so that in case some of the accessions are lost from the field gene bank, we still have a source of these accessions.
This is clearly an expensive operation, so how is it paid for?
It was initially funded by IDRC, International Development Research Center. Now we have another funding, it's a government funding actually, so they saw the need for this in vitro gene bank. They decided to fund this so it could be continued. But this funding will be only up to next year. Then after that, I don't know if the center can maintain it on its own, because it would need much more money than could be appropriated for the center.
There are thousands of banana varieties. Here they have the resources to cope with just 200, and even the funding for this is a hand-to-mouth affair. Other gene banks have even worse problems. One of the world's largest collections of onion and garlic germplasm is held at the research institute in Olomouc in the Czech Republic. Both crops need to be grown out on a regular basis, as onion seeds don't store well and garlic only reproduces vegetatively.
This material comes from Uzbekistan. We collect it in the valley of River Pskem. It is endemic species, Allium pskemense. And for us, this is important because of crossability with onion, and it may be used for broadening the genetic basis of onion crop.
In the disastrous floods of 1997, the entire institute and farm were submerged. Much of the collection could have been lost.
We have more than 7 days underwater. We have flooded our experimental fields. We harvested this material, and we have exact possibility to maintain this material in totally dry conditions in this storage. So I hope that we haven't lost any material from this reason.
Only a few lines were lost, but without in vitro backup, the whole collection is at risk from such events. With state support in the 30s, Vavilov's institute eventually had over 50 separate field stations in different climatic zones. But adequate funding has now been a problem for many years. The seed collections here are still kept on little more than a care and maintenance basis, without proper cold storage facilities.
Now, main task of our institute is long-term storage of our unique genotypes. Each year, we need regenerate about 100,000 genotypes. It is very expensive work. About 3.5 billion rubles we must spend for this process.
The effort needed to constantly regenerate seed samples costs time and money. But the Institute has little alternative if they are to keep their samples viable. And if samples do deteriorate, there is no going back. Many of them are now extinct in the places where Vavilov collected them. In 1995, an emergency grant was provided by the United States Agency for International Development, so that part of the collection could be stored in these fridges. But without enough money or proper modern facilities, the whole unique collection is at risk.
Recently we presented to the Ethiopia 150 Ethiopian landraces. Ethiopia lost them completely, and they were only in our collection. And we collect all these Ethiopian landraces and returned in Ethiopia to Ethiopian government. But we have many difficulties because we have no money for the studying of our collection. We have no money for the organization of deep scientific investigation in the field of genetics, plant physiology, biotechnology, molecular biology, and so on. All scientific institutes in our country now have a very difficult situation, and practically a salary, middle salary of our professor or of our senior scientist, it's about $50 or $60 per month. And people work in this very hard situation, and people work practically without money. And I'm sure that we can support of our genotypes, and we can support of all collection of the Nikolai Vavilov.
The Vavilov Institute has survived on little but the ingenuity of its staff many times before. One policy is to allow staff to have their own private plots on the research farm at Pushkin. Here they grow vegetables to eke out their meager salaries, and they also grow out samples for the gene bank in their own time.
In my opinion, the most appropriate way to improve the financial situation here is by cooperation, particularly international cooperation. We've already got some interesting joint projects which do have proper financial backing. One example is a three-way agreement we've got with the International Plant Genetic Resources Institute, Cornell University in America, and the International Potato Center to do research on potato.
Further away from St. Petersburg, most of the outstations have faced even bigger problems as the Soviet Union disintegrated. These collections took dedicated scientists decades of work to assemble, and they will be needed for generations to come. In some places, entire institutes with their collections lie destroyed and abandoned. This is what remains of the Gene Bank and Research Station at Sukhumi in the self-proclaimed state of Abkhazia in the Caucasus. In fact, there are few places around the world where the conservation of plant genetic resources receives the attention it merits. Even the international gene banks are constantly subject to budget cuts and short-term funding. In 1996, an international technical conference at Leipzig, Germany, agreed a global plan of action which was adopted by over 160 countries. There is now official acceptance of our global interdependence. But will this be translated into action? And if so, who will bear the cost? One thing is certain. If we do not act soon to rescue and protect the Earth's endangered plant genetic resources, the prospects for future generations will be increasingly uncertain.